Alright, so on to our third section on DNA structure and function, and next up we're going to be taking a look at transcription. Alright, so transcription translation and DNA replication, which we'll see in the next chapter, all involve three main steps. There's initiation, elongation, and termination. So the nice part about these three really big important processes is at the very least they all have the same names of the steps. Right? So with initiation, we're going to be starting the process. And so with initiation of transcription, what's going to happen first is the DNA double helix is going to be partially unwound in the region of the gene that we're going to transcribe. So again, what's important to remember here is we're not transcribing the entire molecule, and we're certainly not transcribing the entire genome. We're just going to do the gene that we're going to use right now. Right? So that's kind of the key thing. So at the beginning of the gene, we're going to partially unwind the DNA double helix, not permanently, but temporarily. And so what needs to happen next is that RNA polymerase is going to bind to what's called the promoter region on the template strand. Now the template is the copy we're going to use. The promoter is like the start signal. It's the beginning point for the gene. It's where the whole process is going to kind of kick off. RNA polymerase is a very important enzyme. RNA polymerase is an enzyme that builds RNA. And so when you see the term RNA polymerase, you should immediately think, okay, this is going to be transcription. This is not translation. This is not replication. Polymerase means transcription. Okay? So the next step is elongation. And what's going to happen next is that RNA polymerase is going to synthesize a new strand of RNA based on the DNA template strand. Okay, so RNA is synthesizing synthesizing RNA using the DNA template strand. And that's kind of our next step. Now, the differences between RNA and DNA are important here because we are going from DNA to RNA. So we have to keep those in mind as RNA polymerase builds that growing strand of RNA. The bases in DNA are going to be complementary to the new ones that are being created in the language of RNA. So when you think about what transcription means, if you're transcribing something, essentially what you're doing is copying it. So we're going to copy, in this case, from DNA to RNA. We're still within the same language, more or less, of nucleotides and nucleic acids. So we're going to copy from the DNA language to the RNA language. And so the RNA will be complementary to, or have the opposite bases from, DNA. So if DNA has the base A, RNA has the base U. If DNA has the base G, RNA is going to have the base C, and so on. Okay? The bases are the opposite for each of these two molecules. As this is kind of progressing, as the RNA gets bigger, the growing RNA strand is going to begin to peel away from the template strand and eventually kind of go off in its own direction. Right? And so the other thing to remember here is that DNA, which was partially unwound, is going to rewind itself after RNA polymerase has kind of moved down the DNA along the gene. Okay, so now we have in this picture, we have our DNA double helix. We have a template strand down here on the bottom. This orange thing is RNA polymerase, and it's creating a strand of RNA complementary to the bases in DNA. The last step in transcription is going to be termination. So once RNA polymerase reaches what's called a termination sequence, that's where everything is going to be kind of completed. That's where things are going to be released. So at the end of termination, we're going to get several things. We're going to get our original DNA double helix back the way it was, completely the same. We're going to have our RNA polymerase released as well, because we know once enzymes are used, typically they're going to be recycled afterwards. So RNA polymerase is available to transcribe another gene somewhere. We're also going to have the newly completed RNA strand, which is kind of the whole point of doing this, is to get this RNA strand, which can then go off and be part of translation, typically. So this RNA, again, could be messenger RNA, could be transfer RNA, or it could also be rRNA, depending on the needs of the cell at that point in time. Okay, so our three steps: initiation, RNA polymerase reads the DNA template strand and creates RNA. We have elongation, which is where the RNA is actually being created, and termination, where RNA polymerase comes off, DNA is released, and the newly completed RNA strand is released as well to take care of its job. Now, in eukaryotic cells, there's a series of modifications which need to happen next before the RNA is ready to leave the nucleus, and primarily this is going to correspond to mRNA, or messenger RNA. Right, so one thing that needs to happen is that it turns out the environment of the nucleus and the environment of the cytoplasm, where the RNA is going, are fairly different. And so the RNA molecule needs to have some protection on both ends so that the ends of the molecule don't get degraded once they get out to the cytoplasm. So we have to protect both the 5' end with what's called a 5' cap, and we also have to protect the opposite end, the 3' end, with what's called a poly A tail. Right, so we really haven't talked yet about what 5' and 3' mean, but for right now, we're just going to say a cap goes on what's the 5' end, and a tail goes on what's called the 3' end. Tail goes on the 3' end. Right, so you can think about the 5' cap and the 3' tail as being kind of like those little plastic things on the end of your shoelaces. They protect them. They protect them from fraying, they protect them from damage. If you are familiar with Phineas and Ferb, you know those are called aglets. Those little plastic things on your shoelaces are called aglets. So they're going to help protect the ends of the molecule from damage once the RNA gets out to the cytoplasm. That's a really important couple of additions there. The other thing we have to do is we have to get rid of any non-coding sequences which might be present in the RNA molecule. So it turns out that sometimes in DNA, there are little, or in some cases not little, big stretches of DNA which don't actually code for a protein. They're there, but they're just kind of in the way. They're like commercials that get in the way of the rest of your show. So we have to get rid of those non-coding sequences so that they don't get turned into protein along with the rest of the RNA molecule. The non-coding stuff gets thrown out, and the coding stuff gets pieced behind to form an actual translatable piece of RNA. Okay, so at this point, the RNA is going to leave the nucleus, it's going to go out to the cytoplasm, and it's going to be translated. Okay, so another thing that we see in eukaryotic cells that makes us actually less efficient than prokaryotes is a lot of times some of these more complex biochemical pathways might have 10 or 20 steps, and each step might require an enzyme or two. In a prokaryotic cell, all the enzymes you need for some function are together at one spot on the chromosome. In eukaryotic cells, that isn't necessarily the case. You might have enzymes spread throughout the genome for the different enzymes you need for some biochemical process. So enzyme 1 is on chromosome 2, enzyme 3 is on chromosome 17, enzyme 4 is on chromosome number 7. They're all over the place in the genome, which makes them a little bit less efficient. The other thing I mentioned in the last slide as well is getting rid of those non-coding sequences. So the coding sequences, the ones we want to keep, are called exons. And the non-coding parts, the parts we want to get rid of so they don't become part of the protein, are what are called introns. Okay? So the way that I remember this personally, I don't know if this works for you or not, but the way I remember it is that introns are in between and exons are expressed. Exons are expressed, introns are in between. Exons are turned into protein, introns are cut out, thrown away, and not turned into protein. So we have to get rid of the introns and then piece together the exons. Okay, so when we're done with transcription, initially before the RNA leaves the nucleus, we've added the cap, we've added the tail. Now we have to get rid of all those extra introns. And so there's a whole set of enzymes in the nucleus to help us do that, to make sure we cut out the proper introns and do what's called splicing together of the appropriate exons to make it what's called a mature mRNA molecule. And at that point, the mRNA does leave the nucleus, goes out to the cytoplasm, finds a ribosome, and that's where we're going to see translation beginning. Okay, so here's kind of a quick look at what's called splicing, which is getting rid of introns and piecing together exons. Up here we have DNA, so we have kind of the part we're not transcribing here. We have our first exon, our second exon, our third exon, and then some more DNA, which we're not going to transcribe. Between these exons, though, there are two introns. These are like commercials that we don't want to keep, so we're going to throw them away. Right, this is kind of like back in the old days before DVRs were super common. The first real DVR, I think, was called TiVo. And one of TiVo's claims to fame was you could actually record your TV show and program it to leave out the commercial. So it would stop recording before the commercial and begin recording again after the last commercial, so that when you got home after work and turned on your TiVo, you could watch your show minus all the commercials. That was the idea here. Now, what this is doing is we're getting all the good stuff, the parts we want to keep, piecing those together, and getting rid of all the commercials in between. So those are the introns we're going to get rid of. All right, so once we transcribe this, we get a piece of RNA, exon, intron, exon, intron, exon. We're going to add a tail to the 3' end, we're going to cap to the 5' end, and then we're going to splice together all the exons. We're going to cut out the two introns, splice together the 3 exons. That creates this mRNA here, which is now mature. It can leave the nucleus through nuclear pore, go out, and be translated. Okay, so another look at how splicing works. We take DNA. Initially, once we transcribe it, we have both exons, which are the yellow ones, and introns, which are the purple ones. We've added the cap, we've added the tail. Now we got to get rid of these purple things. So we're going to get rid of the introns, and what we're left with is a mature mRNA molecule that's just made of exons, a cap, and a tail. And that thing is now ready to be translated. Right? Now it turns out, and this is something which we're just learning more and more about now, is that a lot of genes can go through what's called alternate splicing, which is where you can actually get multiple protein products from a single gene. It used to be thought for a long, long time that each gene could only give you one protein. And so as it turns out, that's not necessarily always the case, because if you take a gene and splice it in different ways, you get different products. So if you were to leave in an intron, for example, or take out an exon, for example, things you wouldn't normally do for the gene, but if you were to do those things, you would actually create a different protein product. So you can start with a single gene and actually get 2 or 3 or 4 or 5 different proteins out of it. A lot of human genetic disorders are the result of alternate splicing. It's not so much that the gene is the problem, it's that how the gene is transcribed, which is the issue.